afternoon and greetings of the day to all participants, and thanks to organizer who provide me this opportunity to sharing my views present over here. But I would like to say that just my topic for this today's presentations is somewhat different from the previous speakers, as I am from the pharmacy field and I am a formulation scientist. So my main focus for this presentations is on the formulations point of view. So today I'm going to present over here related to the targeted nanoparticle approach for the treatment of lung cancer. On this topic, our team is still working on. So as we know, cancer is nowadays, cancer is become a big threat to human beings, and nations will add continuously new cancer patients every year. And we can say it is not a one disease, but it's a, we can say clusters of a many diseases. And as per one reported, it shows the death from the cancer worldwide are projected to continue rising with an estimated 12 million cases in future by 2024. So still it is an alarming for us. And once the just you have to see the divisions of a cancer. So cancer cell divisions is rapidly mutate, and once it will be passed from all the mutation stage, and at that time it's convert into the uncontrolled growth. And at that time, it is very difficult to manage with the cancer disease. And out of all the cancer, our team is choose for the working on a lung cancer, because as per the data even so we have to see that lung cancer is the primary cause of cancers among the various cancer, and main it's due to the smoking, habituate of smoking, mainly people with that. And if the patients who are suffering from the lung cancer, so just you have to see that their airway become very obstructed. So it is then challenging for the researchers as well as for the F&D scientists to deliver the drug through the obstructed airway up to the targeted sites at the lung. Now we have to see just the anatomy of lung, because when the person was suffering from lung cancer, their airway, or we can say alveoli, it is the end part of a lung, it become damaged. So again, it is a challenge is when need to reach the formulations to the alveoli by passing all these 23 stages. So it is a main challenging behind the development of targeted formulations for lung cancer. And as such, we have to see key treatment option for lung cancers are available: chemotherapy, radiations, and surgeries. And as we all are aware, each and every treatments have some side effects who produce the adverse effect to the healthy tissue. So treatment options. So all the treatments, we have a problem. Those by this common treatments, the effect to the healthy tissues are seen more. So that's why patients are feeling some severe side effect. So apart from this, some chemotherapy and all that thing, we are working on a inhaled chemotherapy. So inhaled chemotherapy have some advantages, along with this some challenges are also there. By just we have to target the dosage form to the particular site with reduced time, reduced dosing frequency, as well as improve the viability followed by patient's compliance, but also have some challenges. So those challenges we have to fulfill by the development of some novel formulations, or we can say do the thoroughly working in a F&D form. So for the nanoparticle point of view, mainly we have to deal with the aerodynamic particle size along with the physiological components. So for that, whenever we have to deal with the aerodynamic particle size, nanoparticles is an attractive therapy due to the flexibility for the development of size. It is also suitable for the potent drug with high encapsulation efficiency, as well as the targeted and controlled delivery. So for that, out of some various pharmaceutical nanoparticle formulations, liposomal, it is an best options, or we can say it is an alternative options for the development of nanoparticles due to their excellent release behavior, release at the particular targeted site, excellent biodegradability, as well as most important thing, they were able to incorporate both hydrophilic as well as the hydrophobic drug molecule. And most important thing, liposomes can be surface modified. So we were able to entrap with multiple functionality for the long systemic circulations as well as the accumulations at the targeted site. Now, along with the nanoparticles, we have combined with the magic bullet concept, that is the concept of a targeted. So various targeted drug delivery systems are used for the various benefits, including the improve the efficiency, side effects were reduced, followed by dose or dosing frequency. So ultimately, it will improve the patient's compliance. But still, the targeted drug delivery have some challenges, because most important part for the challenges is difficult to retain at the targeted site for the sufficient period of time. Also, physiological barriers are there for the reach the formulations at the particular targeted site, and obviously safety point of view is also important. So for that, various challenges are also there. And if we have to see in the level of targeting, so mainly 3 levels of targetings are there: primary, secondary, followed by tertiary. And each and every targeting have some challenges. So for we can say for the primary, retention is challenges. For the secondary, delivery as well as retention is challenges. And even for the tertiary, all are challenges, including the viability even also. So by taking a view of this targeting approach, mainly for the pharmaceutical formulations, 2 targeting approach were developed or were applied: passive as well as the active. Passive targeting approach is depend on the circulation's time of formulations through the body. Ultimately, if I am say, it is totally depend on the particular physiology or anatomy of that particular cancerous tissue. While active targeting approach, it is a more favorable because it's work like lock and key phenomena. If we have to apply active targeting agents, then at that particular receptors, once they will reach at that particular receptors, then and then it will be give the release effect. So if we have to combine this approach, liposomal, or we can say any nanoparticle formulations with the inhalations form along with the active targeting approach, so might be the formulations were reached at the lung targeted only, rather than release or rather than reach to the healthy tissue. And even though it's proved that the administrating the nanoparticles via IV route may result up to 5% of nanoparticle reach to the lung, compared to 3 to 14 times higher of nanoparticle concentrations when administered via the inhalation route. So by taking this concept, the how to apply those concepts for our study. For that, we have to select a drug, silibinin. Silibinin have an effective anti-cancer activity in lung cancer. It is already proved. And we have prepared a nanoparticle, prepared a liposomal nanoparticles, and here folic acid, folate, we have used as an active targeting agent. So our compositions of nanoparticle, that is the folate PEG DSPE, that is the folate is active targeting agents, followed by cholesterols and lipids, prepared by the Lipex extruder, followed by thin film hydration methods, and by the extrusions technique. And now, as a formulation scientist, we need to more focus on a statistical parameters of formulations, because whenever we have to formulate the formulations in our lab, there are various factors which are highly affected on the quality of our formulations. And those factors, we need to rectify it for getting an optimized, effective formulation. So as a part of a DOE means design of experiments. So as a part of DOE, first we have to apply the Plackett-Berman design for the screening purpose. For checked out which parameters are highly affected on the quality of our formulations. So we have to select the various parameters related to the formulations like cholesterol ratio, chloroform methanol ratio, organic solvents, volume of aqueous solutions, evaporation speed, as well as the water bath temperature. These all factors which are related to the formulations as well as the process variables. And we have to focus mainly on the particle size and encapsulation's efficiency of a nanoparticle. So by applying this design, we have to prepare total 18 batches with the different compositions according to the design, and design is run by the design expert software and check the value of particle size and encapsulation's efficiency. And through the statistically as well as through the some analytical, we came to know about that out of 7 variables, including processing as well as the formulation, lipid cholesterol concentrations, as well as the sonications time, these 3 variables are highly affected on the quality of our formulations. So those 3 variables which we have to select it for the further study, and again we have to optimize by different DOE design, that is a design of experiment by Box-Benneken design. Box-Benneken design, it is a very renowned design which are used by the data scientists for the optimization purpose, optimized of the formulation. The same, this design we have to run by design expert software. And this is a layout of design by Box-Benneken design. Experts give different 15 batches along with the different composition, and same for all the batches we have to study about the particle size, encapsulation efficiency, as well as the drug release phenomena. And later on, for the further study, we can check the relationship of all the components against our formulations. For example, I said for the particle size, cholesterol concentrations, as well as the lipid concentrations, it shows the positive effect. It means if my lipid concentrations and cholesterol concentrations was increased, particle size was increased. And for the sonications time, it shows the inverse relationship. If sonications time was increased, my particle size was decreased. So by this way, we were able to getting about the one optimized formulations by taking and concentrations on the different formulations variable like particle size, drug release phenomena, as well as the encapsulations efficiency. So these are the, we can say it is a contour plot and 3D response plot. Both plots are helpful to summarize about the effect of data. And after that, we need to do the validations of design, whatever we run through the design expert software. For that, we have taken one overlay plot, and through this overlay plot, we came with one optimized concentrations, which having a compositions of 0.3% lipid, 0.7% cholesterol, as well as the 7 minutes is sonications time. This is our ideal compositions, and through this compositions, definitely we came to know, came to getting about the formulations with our required response. including the particle size, release behavior, as well as the encapsulation's efficiency. And same thing we have to check over here by calculating the error. If error is more than 8%, then it indicates the design is not validated properly. But in our case, error is less than 8%. Hence, we can say we are getting the perfect optimized formulation. And these are the different phenomena or different physicochemical evaluations of all batches, including the PDI, that is the polydispersibility index, drug content, yield, as well as the zeta. And by the release behavior, in vitro drug release, we have to done by the USP dissolutions apparatus type 2 by using an cellophane membrane with the dissolutions media of lung fluid. So by the in vitro drug release study, we came to know about that our formulations shows the release behavior from 60 to 80% within 9 to 10 hours. So might be we can say it's give the sustained release phenomena by taking an in vivo. And again, for the confirmations, we have to go with the NMR study for checked about the whether folic acid is properly conjugates or not with our liposomal nanoparticle. And by this NMR study, it shows the folic acid both successfully conjugations with the liposomal nanoparticle through the PEG backbone by the methylene group. And same peak is reflected in this graph even also at 7.21 ppm. This is the data of scanning microscopy, and it also shows that key particles were spherical in shape even also, and does not have any obstructions were seen on the surface of nanoparticles which are conjugated with the folate. And most important thing behavior, that is the aerodynamic behavior, because ultimately our inhalers is directly work with the aerodynamic behavior. So aerodynamic behavior study we have done by the cascade impactors instruments, and even though by seeing the data we came to know about the maximum amount of drug which is reached at stage 5, 4, 5 and 6, and it's indicate that our formulations have a major chances to reach at the deep part of a lung such as alveoli rather than reach at the origin of a lung. Same we have confirmed by the all the parameters like MMAD, mean mass aerodynamic diameters, as well as the geometric size distributions. So this is the witness of our aerodynamic behavior. The formulations was successfully reached to the deep part of a lung. And here we are working for the lung cancer. The same formulation cytotoxicity study were done by using a A549 cell line, which is a human lung adenocarcinoma cell line, and it shows the inhibit of growth, or we have to successfully see the cytotoxicity study for our formulations, and IC50 value for our formulations is nearer about the 26 microgram per ml, and check against the standard drug for the lung cancer, that is the paclitaxel. And same thing we have to confirm by in vivo in a Vista rat. So very first we have to develop the lung cancer in Vista rat by using a NNK. NNK, it is a cancer inducing substance. So it's a protocol of 8 months. So until continuously for 8 months, we have to work on the NNK model. And for this, we have to design of MDI and deliver those calculations For delivering our formulations to animal, we have to use the microspray aerosolizer. And before using into the animal, we have checked that efficacy of percentage delivered. And it shows near about the 99%. So it shows about the successiveness of our lung cancer model into the animal. And during the development of lung cancer, we have checked about the body weight analysis, food intakes, as well as the water intake for the rat as a supportive study to confirm about the growing of lung cancer as well as the support of cancer-inducing substance in the rat. And finally, after 8 months, we have gone through the histopathological analysis for the lung, or we can say for the organ of animals, to check about the, or to confirm about the development of lung cancer. And through histopathological analysis, it shows the lung cancer were successfully developed into the animal. After that, we have to given, or we have delivered our formulations to the animal up to 3 months, along with take a reference of a marketed formulations, including the paclitaxel IV. So by this way, even also, we came to know about the successfulness, or we can say we have to see the remarkable difference or remarkable efficiency of our formulations against the developed lung cancer model. Same, it is witnessed by the tissue distribution study in red after the inhalations administrations after 24 hours, and it shows the maximum drug concentrations it presents in a lung rather than a different organ, followed by kidney, liver, as well as the spleen. And same, we have to confirm with the IV administrations, and even though we have to see by the IV administrations, they have a less chances to deposits, or we can say to reach the formulations to the lung rather than the organs of a liver, spleen, kidney, as well as the maximum amount was reached in a plasma. In vivo bioavailability study compared with the solutions and also compare do the analysis of plasma and lung kinetic, and it shows the AUC, that is the area under curve, was increased by 21 folds after administrations of nanoparticle formulations. So it's indicate it's not increase the viability only, but it also facilitate the long-term retention even also, so which might be useful for sustained release effective treatment. So same formulations we have to put for the accelerated stability study as per the ICH guideline for the 6 months. And by this accelerated stability studies, we have not seen any major differences in a particle size, because main challenges for the nanoparticle formulations is to manage the particle size, because during the storage might be aggregations where start. So once the aggregations were start, then definitely it should be converted in a cluster form. So it's create a problem related to the formulation stability as well as the effectiveness. So it's proved that there are no any issues related to the stability study. And once the formulations were developed, we have to fill the formulations into the MDI and checked about all the physicochemical parameters related to the MDI. Specifically, right now we have to target, or we have to focus on the drug content as well as the uniformity among each acutations of a nano formulations, because still work is going on related to the connections of aerodynamic behavior along with the anatomy of lung with the inhalations technology, including some modern novel drug delivery system. So by this way, we can say there are various nanotechnological approach were used to treatment of lung cancer, not only liposomal, even though we are going to use polymeric nanoparticles even also. And as a formulation scientist, this is a DOE, or we can say QBD approach, which are successfully used to come to know about the one optimized formulation, then size distributions. It is also important, which we have to investigate by cascading factors, and it's indicate the deep targeting of formulations to the alveoli, as well as the when we have to compare with the free silibinin, the silibinin loaded with the folic acid conjugated, it shows the higher deposition in a lung tumor tissue along with excellent activity. And same, it is witnessed by tissue distribution study and histopathological study, which we have to done in a rat. So finally, we can say this targeted drug delivery approach exhibit a wonderful approach for improve the therapies for lung cancer, and experimental nanoparticles for the pulmonary delivery may have a considerable pharmaceutical potential for the treatment of lung cancer, and might be in a future era. It shows the inhalable conjugated nanoparticle may open a new avenue for the efficacy treatment of lung cancer, mainly for reduce the side effect. Those side effects commonly we have to see in a normal healthy tissues, because by this way our formulations were divided. So if the formulations is in untargeted form, then dosing frequency were increased, as well as the dose frequency or side effect we have to see commonly in the whole body part, including the healthy tissues. So thank you very much. And I belong from the Department of Pharmaceutical Science, Saurashtra University in India. And in our department, various approved courses are there, including B.Pharm, M.Pharm, PharmD, as well as the PhD. And here our research team is working on mainly formulations development along with the collaborations with some industries.